So, hello everyone. Thank you for joining our first webinar. We'll have more and more webinars. Let me introduce the ones on the call. It's me. My name is Stan. I'm chief executive of MyLabs. Back many years ago, I was one of the initial creators of MyXCR software. I guess MyXCR is why majority of you, I mean our attendees, joined this webinar. On the call, we have Dmitry. Dmitry,
Hello,
please.
everybody. I'm a CTO in my laboratories, and yeah, so all the technical questions, I will be answering them. And yeah, I'm one of the developers of MyXCR,
And Platforma.
and Platforma as well.
Yeah.
That's true.
And Mark.
Yeah, hi everyone. My name is Mark. I am the head of the customer success and customer support at My Laboratories. I have a very deep experience in academia, in molecular biology and immunology. So I'm in the company. I'm the one who's responsible in like communicating with clients and helping them to achieve whatever they need to achieve with using our software.
Clients, customer support, success. We are, first of all, scientists. At least for many years, we were doing science before we switched to the thing we are doing right now as a company. And the topic of our today webinar is a scientific topic. So I will start with just sharing my screen and going, well, we'll see how it goes. So let me share my screen. Mark, Dmitry, tell me whether you see my screen.
Good.
Yeah, everything
Seems
works.
good.
Yeah.
Amazing. So the topic for our webinar today is a very scientific. It's actually related to the T-cell repertoire data analysis. And with our new software, it's called Platforma. We are going to reproduce some of the results published in this research paper related to the different age patterns in the repertoire repertoire data, T-cell repertoire data, using Platforma software. So first of all, I know many of you use MyXCR. It's a command line software. For many years, we were building a new software called Platforma, which is aimed to enable biology people who are working with planning experiments and generating all the data, to enable that people get answers and get insights from the data themselves easily. So with this specific webinar and with this specific publication, we will reproduce the main results showing that the diversity of the immune repertoire, like you see on this plot in Platforma, goes down with the age, or more advanced results showing, for example, what's called in the paper a homeostasis of the repertoire, showing that the public clonotypes are prevalent in the elderly people, and many other results. So the main idea of Platforma is to enable people, scientists, biologists, to get all this results easily, just with buttons and clicks, and do it independently and really quickly. So during this webinar, I'll just go step by step, starting with like the very first steps of the analysis, the FASTQ files, and navigate how to get and obtain all these results yourself in Platforma. And Dmitry and Mark will help me because I will ask questions to them. Both of them are authors of this publication and several other publications which we use for this webinar, and they know the software very well. So guys, Dmitry and Mark, please also help me. Interrupt me if I'm saying or navigating into a wrong direction, okay?
Sure.
Will do.
Okay. So let's go to the main screen of Platforma, to the main page of Platforma. Even before that, when you just start the Platforma software.
And sorry, by the way, you can type in any questions during the presentation. Me and Mark will answer them, or we'll
In the
or
Q&A,
in the
yeah.
chat, or yeah, or we'll tell something
Oh,
in the
chat
end.
is actually a separate thing, right? We have Q&A and we have
Yeah,
chat.
Q&A. Yeah, I mean in the Q&A. Yeah.
Okay.
Yeah, please do that. okay? If you see an annoying Zoomas is accessing your screen, this says all the questions to Apple. Many people complain. Okay, so let me show you what you see first when you run the Platforma. So the first screen, when you just download Platforma app from our platforma.bio website, the first screen that you see is this one with the way how to, well, with the question where to run the analysis. Well, we are talking about next generation sequencing data and analysis of NGS data, that's large data, and obviously to process this data, especially the kind of data that we have in this study. There are a lot of samples, gigabytes of sequencing data. To process this data, you need some infrastructure, server or something, which in the majority of academic institutions or commercial companies are always exist. But even if you don't have any computational power, you can run Platforma and all the analysis just on your personal computer using this built-in feature. Or if you have, like many people have in their labs, like some SSH access to a server, then you just put the credentials to your SSH server here and connect, then all the analysis, all heavy work will be done on that remote machine. We are going to use the server that we have somewhere in, I guess, in Hessner in Germany, yeah, to run this heavy calculations. And I'm connecting from my desktop app, which is on my computer, to that server. So here we have a list of projects that I have in Platforma. The mice aging project is the project that is actually we're going to reproduce. And starting from the very beginning, I'm creating a new project, webinar TCR. Great. And we have empty project. The universal entry point in any project is actually samples and data. And when I click this add block button, you see a gallery or a registry of available applications for very different types of the analysis and available in Platforma. And by the way, MyXCR is a tool for T-cell and B-cell NGS data analysis, but Platforma is a tool for any kind of NGS analysis. And we have applications for gene expression analysis, for single cell, for, I don't know, other things here. And more such applications, we call them blocks, are coming, some of them developed by third-party bioinformatics scientists from academia or industry. Yeah, that's the idea. That's actually a platform. So the first block I am adding is samples and data block. This is the app or block allowing to import raw sequencing data into the project. And there are two ways how you can do this. You can either select some FASTQ files located on your computer, which is not the case for this study. The data is really big. It's located on server. Or you can instruct a Platforma to have access to remote storage. And we have the storage configured here. And import the FASTQ files located on that remote storage into the project. So I'm going to select all of the FASTQ files corresponding to this study from the remote storage and import them into the project. And you see this app, this block samples and data automatically recognizes the sample names from the file names and the read one, read two structure of the data. So the pairs of read one, read two. So this specific data, this is the data where the amplification of alpha chain and beta chain was performed separately. So we have a separate set of files corresponding to alpha chain and a separate set of files corresponding to beta chain. And we can import them all. It's not a problem. Let's say we call this data set TCR alpha, and then we filter files that we're going to import using this pattern, including alpha. So everything before this will correspond to a sample name, and everything else, like read one, read two here, etc., etc. So that's created. Done. So we have our samples. For every sample, we have read one and read two. But before clicking the run button, let's also add TCR beta files here. So I'm going to do this, TCR beta, yeah, and do actually the same, just plug in beta here and import same samples. So the sample names are the same, but there are two data sets, TCR alpha and TCR beta. So here in the metadata section, you actually see this. For every sample we have, every sample belongs to two data sets, TCR alpha and TCR beta, and we can import a metadata corresponding to this project as well. Let's say we have a metadata in Excel or TSV or CSV, then we can import absolutely arbitrary metadata associated with our samples using this import metadata feature. Yeah, and now we have all our samples. For every sample, we have different metadata. Well, the most important for this specific study is the age of the mouse, age in weeks. Yeah, and that's it. Let's click run. And with this run, this app and Platforma will just import the FASTQ files into the project and make them available for the downstream blocks. And now the next step, the basic step we want to do, we want to get the clonotypes, TCR clonotypes. And for that, we have a MyTCR clonotyping block in the registry, which we are going to add. And basically this block just runs MyTCR tool. Yeah. So let's first do it for TCR beta data set. Yeah, and by the way, very important thing, we added two data sets here. But as I mentioned, Platforma is not only for B cell or T cell analysis. We have gene expression, other things here. So you, for example, may have same samples, one data set for VDJ data and another data set for gene expression, single cell gene expression data. Yeah, and then you can analyze single cell combined VDJ plus single cell data, integrated, etc., and do a lot of things, which we will cover in our subsequent webinars. But now we're using this nice study to get some interesting results and visualizations. So this is MyTCR clonotyping block. The settings are really simple. You just need to take one of the presets, like you do in MyTCR, corresponding to the library preparation protocol that was used to generate this data. And we support all types of chemistry, Takara kits, Kaigen kits, Illuminampli-Sec kits, 10x Genomics VDJ kits, parts by whatever, everything is supported here, and you can just find the preset if your data was generated with some of the vendors, or if you use a custom library preparation protocol, which is not the one provided in the presets, then you can also use a special, what we call a preset in MyTCR. It's just a file with proper MyTCR parameters describing the architecture of this library. And in this specific study, the library preparation protocol was custom. So we are going to use a custom preset with MyTCR to analyze this data. What else here? Additionally, this is TCR beta. So by default, MyTCR will analyze and extract all immunoglobulins, alpha, beta, and gamma delta chains. But in our case, we can just leave here TCR beta. Since we know that the library was simplified only for TCR beta chain, then that's it. And there are some advanced settings. So if you want to go deeper, there are advanced settings. One is very useful, and we always recommend using it, is this one. Take only this number of reads into the analysis. So for the test run, to test that everything works, you don't want to spend several hours, especially if the project is big, on the clonotyping, on the upstream analysis. So you can just plug, let's say, 100,000 of reads here, then 100,000 of reads will be taken from every pair of FASTQ files, just as a dry run to see whether how everything looks like. Yeah, it's just 100,000 of reads is already good enough, yeah, for this dry run purposes. And that's it. And you run. Yeah. And what's going on under the hood? Under the hood, Platforma will run the tool, the workflow, the pipeline behind this specific block, MyXCR Clonotyping, effectively just running the MyXCR command line tool on the server, and you just enjoy the nice user interface, buttons, visualizations, clicks, etc. For every sample, you have an overview of the quality of the library. And here we certainly see some signals that some alerts, like something is not like ideal or even bad. We can click on the individual sample to go deeper and figure out what's went wrong. Yeah. Or if any problems. Yeah. We see the alignment rate and the proportion of the successfully aligned reads. We see that this sample is really TCR beta. So the majority of the clones are TCR beta with just a small fraction of TCR alpha contaminations. In the quality check section, you see, well, human readable, well, hints what's the problem, what is the potential problem with this data, why there are alerts. Like here, we have 10% of barcode collision in the clonotype assembly. The library preparation protocol used in this study includes unique molecular identifiers, and this quality check tells us that the diversity of the unique molecular barcodes is low, which is a feedback to the wet lab. Yeah, and something may be changed to improve this. You can go also deeper to check the raw logs of the execution from the command line reports, as you typically do this with command line MyXCR, etc., etc. So, yeah, what's next?
Somebody may ask why it was that fast, that the screen like appeared right away with all the results. That's the result of a feature we call deduplication, because as Stan showed in the very beginning, that there is a project where the same data is analyzed with the same parameters. The Platforma knows about the results. So when you analyze the same data with the same parameters, those results are recovered from that project, if it exists on the back end, and shows you it right away without waiting for the computations to complete. So basically, if something is already calculated, you don't spend time recalculating it, which helps with interactive, iterative analysis that in many cases the biological analysis is.
Yeah.
So yeah, continue.
That's really important. Yeah, because the real type of the analysis people are doing is very interactive and iterative. You change something, you rerun, and you want to see the difference. And with this feature, the recalculation will happen only on those parts of the workflows that actually changed. So the next thing we do, well, the basic thing we are always doing, we just want to look at the clonotypes, to look on what we have in the data. And for that, we have a block called Clonotype Browser for VDJ clones. Yeah, and yeah, here in settings, we select the data set that we're going to look at. That's our TCR beta data set, the only one that we analyzed with MyXCR Clonotyping block. Yeah, to analyze TCR alpha, we will add same MyXCR Clonotyping block, select TCR alpha dataset, and run the analysis. So that's our clonotype table. In the first section, we are looking on the individual samples. For example, yeah, those are our samples. We have the repertoire. Here we can, let's say, since the data has unique molecular barcodes, we have this number of UMIs as the abundance measure for our clones. Yeah, we have all the information provided, like depending on the design of the library. This library covers only CDR3 region. So we have CDR3 sequence, amino acid, and nucleotide CDR3 sequence of every clonotype, V gene, D gene, J gene. C gene is also partially covered with this library, etc. Another very useful section here is overlap. That's one of the very powerful features of this Clonotype browser block. In the overlap, we see how those individual clonotypes appear in different samples. Let's say we have same samples, yeah, but now if I sort by the number of samples, so this is already quite a big data. So this sort already takes time. There are, by the way, let's check. Let's scroll down to see how many clonotypes we have in the full project. So we have 344,000 of clonotypes, yeah, in the full project. So it's quite a big table. And if we sort by the number of samples, then we see the clonotypes. Let's say this clonotype appeared simultaneously in 14 samples out of how many? Out of 14 samples that we have in this study. Yeah. So then, for example, we can go to the columns and check, and let's say add the abundance of this clonotype, let's say the fraction of UMIs for this clonotype in different samples. So we can add this for our samples to see what the abundance of this specific clonotype in different samples. We will do some advancing here to get deeper insights about repertoire, but before doing that, let's add some basic repertoire statistics. The basic one we are going to assess in the study is repertoire diversity. We have a dedicated block in Platforma for repertoire diversity analysis, and this block just calculate major immune diversity indices like absorbed diversity, Shannon-Wiener, Chow-1. We can add different type of other diversities like, I don't know, Gini index with different types of downsampling, how we downsample. So when we calculate diversity, it's extremely important to normalize the data for a proper comparison across different groups. And this is downsampling feature. We can do different type of downsampling. The most useful one, well, not the most useful one, but the one we typically use is just a random sampling on the abundance measure that we choose for our clones. The abundance measure for our clonotypes that we're going to choose to assess the diversity is the number of UMIs. Yeah. And that's it. Let's run. So those are a list of metrics that are added by default: observed diversity, Shannon-Wiener diversity, Chow estimate, Chow-1 specifically, yeah, Gini index, and D50 diversity. And we have the result here in the table. So for every sample, we have, let's say, the observed diversity calculated on the normalized data set and sampled to get like the same number of molecules, to obtain the same effectively to the same number of molecules in every data set. Yeah, and then we can go here to the diversity graph to build already some useful, nice visualizations. The default one you see here shows the bar plot with the observed diversity across our samples. If we go here, we call it data mapping of this graph maker tool, graph maker functionality. The data mapping section on the y-axis of our box plot, we have observed diversity by default. Yeah, let's say we want to look at Shannon-Wiener diversity. Yeah, and in the primary grouping of our plot, we have sample. Let's remove it. Yeah, and let's use group from the metadata. Remember, in the metadata, we added group. Yeah, the group of young, adult, and old donors. So let's use this group here as a primary grouping to look at how the diversity behave with the age. And now we see already some useful visualization. We have some useful stuff here. We have our Shannon-Wiener diversity for adult, old, and young. Well, to make it look nice, let's go to the layers to the group and change the order. So the young should be on the left, young, adult, old. I also prefer this color. Yeah. And now we have some nice visualization showing that the Shannon-Winner diversity, we can also add a title, I don't know, a custom title with like Shannon. Well, I always forget how to write.
Winner.
Yeah, diversity as a title of our plot, and we see the pattern. The diversity is less in all donors. What we can also do, we can calculate different statistical metrics, like we can put the overall p-value, like Kruskal-Wallis or ANOVA, on top of our plot. It will be calculated automatically. And we also can add, for example, pair-wise p-values with t-test or, let's say, Wilcoxon tests to compare groups and to compare the repertoire across different groups. And all this automatic. It also uses, by default, the correction method, like, say, when we can choose whatever correction method, just we assume that Bonferroni is the best one. Dmitry, Bonferroni is the best one. Yeah?
Depends on the sample size of the sample.
Your first yes was short and sharp. So Bonferroni is the best. Very nice. And we have a lot of types of visualization. So box plot is only one option. You can build violins, you can build, I don't know, bin dots, and well, different stuff here. All is possible. So that's already this result, this box plot with these groups is already one of the results. Let me just check this paper. One of the main results, yeah, of this publication, showing the, well, here we see the observed diversity on the normalized data, and the age group, they have we use just as not all of the samples, and we see the pattern. So, and here we reproduce the same just with buttons and clicks. We have various other metrics of the repertoire which people typically look at, like, for example, gene usage. Let's add this block for gene usage to calculate some other basic properties of our repertoire data. And we can do this basically in two ways. We can either use just the gene, the V gene, or a specific allelic variant of the gene identified for the clonotype. Let's use just genes. It's basic one and useful one. And the J gene and the abundance will be just attached automatically here and run to get. So it will, under the hood, run the workflow, the pipeline to calculate the gene usage. We have to wait. I think it should be really fast, but meanwhile, while it's calculating, let me open the same thing into in the project where it's already calculated. Yeah, and here it's also calculated already. Yeah. So we have our V gene usage heat map calculated in the block, and let's add some things here. Well, first of all, let's go to the layers and remove normalization. By default, the heat map is normalized, but that's not appropriate for this kind of data. We can also change the color scheme. Let's use this one, for example. And here in the template, add a dendrogram, a hierarchical clustering to the data to see and turn off the normalization, which is also turned automatically for the dendrogram. And we see some nice stuff, the heat map with the genes. If we zoom in, then we will see the gene names and our samples. Yeah, what else we can do here? One interesting thing that we can do here is the following. We can, if we want, let's say, to bring some of the metadata to the visualization, let's say group, we can put this group on the annotation layer to have this grouping of our donors. So we have adult, old, and young, but now I don't like these colors, really. yeah, the combination of this color scheme and this color scheme, and that's what we can change in Platforma. We can define and change the colors to whatever color scheme yeah, we want to bring here. And what I wanted to say, so actually what we see here, that the data is grouped, and at least for the old patients, we see that the data somehow clusters together. The same we can do for the J gene usage, or if we can also go to the individual samples and check the correlation between the usage of V genes and G genes for the individual sample. So on the y-axis here we have J gene, on the x-axis here we have V gene. Let's also remove normalization. Yeah, and that's another source of information for us to understand the patterns in our repertoires. The other example of what we have here, we have, for example, CDR3 spectratype, which allows us to look on the distribution of the lengths of the CDR3 sequence. Yeah, and let's say as a normalization, we are going to use unique molecular identifiers and calculate V gene spectrotype without alleles. Yeah, it will take. It should be fast. Yes, Dmitry.
It should be fast, I guess. Yes.
Oh, fast. It's this data set, as we checked in the Clonotype browser, has 500, almost 500,000 of clonotypes, 500,000. So some data, although we sampled it. So yeah, we have a spectrotype. We have kind of bubble plot with V genes and the length, amino acid lengths of the CDR3. By the way, I'd prefer to use descending order on the y-axis of this bubble plot of this heat map. Yeah, we also have a V spectrotype for we can check the V spectrotype for our clonotypes, the length of the CDR3 amino acid sequence, and the distribution of the genes, of the V genes here on this histogram, or just a CDR3 spectrotype with showing the most abundant clonotypes in this specific sample. Here we're switching between samples. Well, and what we see here actually, it tells us that all of the clonotypes here in this sample, that's a deep repertoire data, are more or less evenly distributed without some significant expanded clones. Yeah, so that's the basic repertoire analysis and the visualizations that we can generate here. We can download, we can export those visualizations, and we can tune them, add labels, and do other stuff to make them kind of publication or literally publication ready. But now let's go deeper. Let's do advanced sync. And with the advanced sync, I mean the following. Let's look closely into the repertoire. Here in this paper, my colleagues, Dmitry and Mark, and other colleagues from the academic lab, authors of this publication, they went deeper and they did this visualization, which is very nice. This plot shows the space, the size of the space in the repertoire occupied by expanded clonotypes, by rare clonotypes, and show this distribution across different age groups. So this is very deep visualization, very deep result. Dmitry, correct me if I'm wrong, but this is a deep result of this study, yeah, showing this.
For him, this is one of the most important results.
Yeah. Let's calculate this. So what's done here? We'll go to the Clonotype browser. Let's take our TCR beta dataset and let's annotate the clonotypes that we assume are hyperexpanded, large, small, or just rare, with an annotation. And we can do this using a very powerful functionality of Clonotype browser. We have, it's called annotations. and add annotations specific to our clonotypes. There are two types of annotations we can do. We will use both. The first one we're going to use is per sample. So per sample means that we will annotate the clonotype in each sample, applying the criteria for the annotation to each sample individually. Since we are working with the abundances here, yeah, the expanded or rare is a property of the clonotype abundance. And abundance is a property of a sample. Clonotype has abundance in sample. It does not have abundance just as is. Like CDR3 lens is irrelevant to the sample. It's just a property of the clonotype. But the abundance is a property of sample. So to annotate such, to make such annotations, we are using the per sample type of annotation. And the first annotation we are going to add, Dmitry, help me, hyper expanded, I guess, yeah? Hyper expanded.
Yeah, hyper expanded.
Expanded. Hyper expanded
Yeah.
clonotypes. And the way how we annotate, we just specify a set of filters to filter the clonotypes which we assume are hyper expanded, and then those filters set of clonotypes will be labeled as hyper expanded. And the filter we are going to apply is fraction of unique molecular identifiers. This fraction is calculated for each sample individually. And for hyper expanded clonotypes, we assume that those clonotypes are hyper expanded, which abundance is greater or equal, yes?
Then like 1%.
Then 1%. Then 1%. Yeah, that's it. So hyper expanded. Amazing. Let's
Yeah,
add
this way, actually, the Clonotype browser is not just a browser thing, but actually an annotation too, that it converts this kind of block to something that produces outputs for downstream blocks.
And with this tool, we will show in not in this webinar, but in others, how you can annotate your clonotypes, how you can track your clonotypes across various samples, conditions, metadata properties, track them as individual clonotypes, track them as clusters, find enriched clonotypes, and do many things. But here for this specific simple but very, very interesting study, we just use this as annotation. The second annotation we are going to add is large clonotypes. Large clonotypes are the clonotypes which are larger than 0.1% of the abundance. Yeah, those are large. And also we're going to add large, then medium. Yeah, medium are the ones which are larger or equal than, Dmitry, I need with your
Another, yeah, 4 zeros.
Yeah, medium, small, and small ones will be fraction of UMIs is greater than
5 zeros. Yeah, no, another one.
Yeah. Yeah. And the rare ones are the ones with fraction of UMIs less than this value.
Mhm. Correct.
Those are rare. Yeah. So now we have our annotations. Very important thing. The order of annotations is important. Those which are on top, they are overridden by those which are at the bottom. So we need to regroup them because the way how we specify those filters, yeah, no, it's correct. So hyper expanded are those which are larger than 1%, large are those which are larger than 0.1%, and then that's it. Yeah. And then if we go to the annotation statistics here, boom, 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 it calculates. Although this is a sampled data, but it's already quite big, so it takes some time to apply all those annotations. And here we see the proportion of those categories of clonotypes in different samples. So we see how much of, let's say, this sample, M11720, each of the categories of our clonotypes takes in terms of the total number of UMIs. Yeah. So that's the data used here. Let's visualize this data. I need to click run here to make this data available for the downstream blocks. And in the downstream blocks, I'm going to add a block called just GraphMaker. So GraphMaker is a generic tool for building visualizations. So I'm adding a GraphMaker tool, and then I'm going to use stacked bar chart, yeah, to build this plot here. On the y-axis, by the way, let me also do the following. Let me rename this annotation to it's called in the paper homeostasis. Yeah, let me change the name of the annotation. Yeah, rerun it. So this label will propagate to the GraphMaker. Yeah, so on the y-axis, we see the space occupied by this category of the clonotypes in the age group, and on the x-axis, we just have an age group. So on the y-axis, homeostasis, on the y-axis, we want to see the total fraction. This is the column coming from our annotations, the total fraction of UMIs occupied by our homeostasis group. So we want, on the x-axis, we want to put the age. In our metadata, this is group, primary grouping. And on the y-axis, as a secondary grouping at the category, we are bringing the homeostasis labels. Yeah. And then let's go to the settings of the stack bar chart and normalize it. Yeah. And we see very, very nice results. Let me just do the following. Let me again bring young to the left. I also prefer this color scheme. It's here. It's not here. It's here. Yeah, I prefer this color scheme. And we see this result. So we see that the space occupied by small clonotypes in the category of young donors is really, really large compared to the old. And in the old donors, we see that the space occupied by medium and large and hyper-expanded clonotypes is larger. So that's the main result, and that's a main result of that specific publication that we reproduced on some data. So we can rename it in the same way how it's labeled in the paper, homeostasis distribution. Yeah, and that's a result, which is really powerful and deep insight that we were able to find with just buttons and clicks. There are other interesting results that we can do here and that we can reproduce from the paper. Let's do one more before we finish. It will take 5 minutes, and then we'll switch to some questions and some discussion. So let me add one more Clonotype browser here. And now let's focus on the clonotypes which we assume are public, which present in more than one, in many patients. So let's do basically the same. Let's select the TCR beta data set that we have here, and let's create another annotation. We call this annotation, how we call it, public clones. And we'll add only one one annotation, rep_pc, and our filter will be number of samples more than or equal than 4, for example. With this filter, we are filtering clonotypes which present in at least simultaneously in at least 4 samples. And what else here important that we use this global annotation option, meaning that we're applying this filter to all clonotypes in the project, not to the clonotypes individually in each sample, but from the whole pool of all samples and all clonotypes that we have, we annotate those which present in any 4 samples simultaneously. We call them rep_pc, representative public clonotypes. And now let's do some other visualization. Let's say again, I can do this another way around. I can just bring GraphMaker under this block with public clones. And that's important thing. On the left side, on the sidebar, there is a flow of blocks, the upstream blocks and the downstream blocks. And the results produced in the upstream blocks become available to the downstream blocks. So that's why I moved public clones up above GraphMaker to make this the results from public clones available in the GraphMaker. And let's use this bar chart plus error bar visualization. We can always change the type of visualization here, but let's use it. And look on the total fraction, total space, total abundance in samples of the public clonotypes. And use H as a primary grouping. Let's change the line plus error bar. And yeah, and we see another nice pattern. So we see that the space occupied by public clonotypes is larger in the group of old of adults and elderly donors, which is another very important result from this study. So basically that's it. That's what I wanted to show with this simple project and this simple basic project. We were able to reproduce, to get, not just to reproduce, but to get some very important results of the study with just buttons and clicks. And this, the whole aim of this is to show how this can be done with easiness by the biological scientists. In the further webinars, we will show how to work with different antibody discovery campaigns and projects, single cell data, including gene expression analysis in Platforma with easiness, and generate many different and interesting results. Meanwhile, Dmitry, Mark, do you have anything to add?
Well, I wanted to add the overall idea of the Platforma that, as Stan mentioned, that there are like not just focus on immunology, that's just a field that we came from, but the general tool for bioinformaticians, basically developers of algorithms and pipelines to deliver their results, their programs, algorithms, and pipelines to biologists for easy application to their own data, to flexible combination of different pipelines and algorithms, because each study is unique and actually there is no like the certain amount of flexibility is required. So, and for that, the kind of the one thing is this UI where you have the blocks. You don't see this direct acyclic graph or something that actually the data, the very simple workflow. So the data goes from top to bottom. That's it. under the hood, all the complicated things are resolved for you. And another important aspect is that those blocks are isolated applications developed by separate developers, very loosely coupled. And all those blocks actually I answered in the Q&A section. So all those blocks that you see, they're open sourced. They are developed using the SDK that we provide for Platforma. And they can be tweaked, or anybody can go and create their own block. And the SDK is developed in such a way, and we are doing it better and better in the easiness of application. So you don't need deep, like, computer science development background to make those blocks. It is not that easy as like clicking, but actually with modern advance in AI, it is really easy to like create the block like this that implements all parts, heavy computations on the back end with some beautiful UI on the front end.
Yeah. So all the blocks that we have currently in the registry, they are just available open source on the GitHub. Yeah. And it's really easy for bioinformaticians to create new blocks. And we have like very different applications so to use Platforma. Regarding the question, Dmitry, I see you are answering.
Yeah, it's better to just answer it.
But answering online, it's super easy to up and running it locally. You just go to the Platforma Bio website, Platforma Bio, you go to the downloads, you download the desktop app for your architecture, Windows, Mac, even Linux, Silicon or Intel. You install it, you run it, it works. If you have
the easiest way to analyze real amounts of data is to use SSH connection. It is like one click away if you have an access to some computational resources. In 100% of the cases, this is SSH. So you just type in your credentials. Platforma will connect. This is like all the data is yours, so Platforma don't call anywhere. So you connect to your server. The backend is deployed on that server for you.
Yeah.
And connection is established via the SSH tunnel. So you can just.
This is what you see when you download Platforma. So this is the easiest way you start, and that's it. By the way, Platforma is absolutely free for academic research. So all academic scientists, they get it for free. Or if you have your HPC access in your organization, in your institution, then you just put the address of your HPC cluster, you put your SSH credentials, like we have this Candy Bobber, and we connect. It will install the backend, all required requirements and everything on that SSH server, and you start working, and that's it. So it's super easy. So let me. Any other questions? We have 6 more minutes. Mark, how do you feel?
Great.
Amazing. So we don't have other questions, I guess, so far. Just want to say thank you very much for everyone joined this webinar. We will share the recording of the webinar and join our subsequent webinars. We will have more and more great stuff here. Thanks. Have a good day and night.
Thank you, everybody.
Thank you, everyone. Have a good one. Bye.
Bye-bye.
Bye-bye.